Let us look at the Griffith's experiment. So, Griffith used basically a bacteria which has two strains. So, these two strains are named as R strain and S strain. So, we should know in the beginning that what is the strain. Okay, so a bacteria, by the way, these two bacteria belongs to the same species and same genus, but they are two different strains. So strains basically they differ with one another with respect to one particular trait. Okay, here the trait is basically virulence. So living S strain is basically virulent and living R strain is non-virulent. So again, maybe if, someone who want to know about these technical terms, virulent means one which has ability to cause a disease. Okay. Non-virulent means it doesn't have ability to cause disease. So virulent bacteria causes pneumonia. So that is why when Griffith injected living S strain into mice, the mice dies. Okay, this is the first experiment. Whereas in a living R strain, so that which is non-virulent, when it was injected into mice, mice is alive. Mice is alive. It makes sense. It obvious, right? Because it's a non-virulent. It doesn't cause a disease, pneumonia disease. Okay. What is interesting is third experiment where Griffith injected heat killed a strain. Okay, this is a dead. Okay, though it has the ability to cause a disease, now it is dead. It can no longer cause a disease. Okay. Now the third experiment is something that is very important for us to understand. Okay, so what is this third experiment exactly? If you look at third experiment, it has both R strain and S strain. So R strain is anyway non-virulent. Okay? So it doesn't cause a disease. So he mixed R strain with the dead S strain, which is heat killed. Okay, when he injected, so mouse again dies here. Okay, so what must have happened? Ideally, since it is there is a strain which is non-virulent and the other one is heat killed, but but this mice died. Ideally, it should not die, right? Because we have a non-virulent strain and a dead virulent strain. Okay, since the virulent strain is dead, it should not cause a disease, but the mice dies here. What must have happened? What must be the reason behind this phenomena here? Why did mice die here? So the inference that we have from this basically, by then we don't know what is DNA, by the way. Okay. So what Griffith called this phenomena as transforming principle. There is something that has been transferred from living R strain to dead S strain. So because of this, basically, sorry, dead strain to R strain. Sorry, I said it opposite. So dead strain to R strain. So it has transformed the R strain into a strain which has the ability to cause a disease now. Okay, this is known as transformation. So there is something that is present in a dead S strain that has transformed the R strain into disease-causing bacteria. Okay, so this is known as transformation principle. But what is that material that has transformed, that has been transferred from S strain to R strain is something that is answered by another experiment, another group of scientists. Okay, but for the time being, this is the experiment.